So again, I want to thank you very much for having us today. It is pretty early over here, but a very great opportunity to collaborate and get to know everybody and everybody's research. So just a little bit more detail about me and the company. I started my education at A&M University with a bachelor's in mathematics and a minor in computer science. After that, I traveled to Boston University, where I got the opportunity to get training in biostatistics and really get introduced into biostatistic analytical methods and epidemiology. After that, I traveled down to Washington University School of Medicine in St. Louis, where I got my master's in genetic epidemiology. And ever since then, my career has revolved around data. So for the last 10 years, it's been data analysis, statistical data analysis, data management. Been very fortunate to work on various biomedical projects, done data management for hundreds of clinical trials when it came to the development of biomedical devices or new drugs being tested. But for the last 3 years, my focus has been on the development of omic analysis. So that's what I'm going to be showing today. The automation of analysis for untargeted metabolomic experiments. A little bit about BioRankings. Um, we've been very successful in our research, in our method development, in our software development, and in part to that is due to the NIH funding. We have over $5 million in NIH funding, and that's part of the work that I'm going to be presenting today. But none of this work could not be possible without these people that I've highlighted today. Dr. Gary Patty at Washington University School of Medicine. We will actually be seeing some data for a batch effect analysis later on in one of the videos. Dr. Shandell at Northwestern University, we work with various people at his lab to help us test and validate the software. Dr. Weinstock, he actually helped in the development of the CART application using microbiome data that you will be seeing later on in one of the videos if time allows. And Dr. Garrett at University of Florida, instrumental in helping us develop this software, and still moving forward, helping us in the new grant that we have recently submitted to the National Cancer Institute to apply similar methods and software to the discovery of cancer biomarkers. So that with that being said, I'm gonna start today by showing you a generic metabolomic workflow and really highlighting the pain points that our software has solved for researchers, and really emphasizing that our software has moved forward with more of an automation component to it. So at the top of this page, you can see a generic workflow that we really like how it illustrates the steps that a researcher might go through when doing their metabolomic research. I'm not going to go through the entire workflow, but you can see you have data collection, some comparative analysis, some manual steps, some database comparisons, some manual steps. Again, really honing in on the fact that, our peers, as you've seen today, are doing great research, great method development, great tool development. But, these tools are great, but, it still requires these researchers to have some kind of computational background, some kind of programming background, and it still requires a user to go in there and spend a lot of time dealing with things like combination or filtration or alignment of features or chromatograms, corrections or comparisons that you finally get to do your comparative analysis. And that's some things that we're going to highlight in the talk moving forward in the later on slides, how these signal processing decisions can really drive the results of your analysis or even drive the next steps in your workflows. So what BioRankings has really done is automate steps 2 and 3, including the pre-processing decisions that go into these different bullet points. And with that being said, I wanted to illustrate and really show a description of what goes into one of these steps. I'm not going to go through this workflow piece by piece, but I want to just get your get you to view what a researcher is seeing. As you guys might be aware, each one of these components requires you to go in there and make decision and parameters decisions. So it's not as straightforward as you saw in the first slide. It's more hands-on. So that getting to that these workflows are time consuming, and it really does delay time to your result. And you will see later on that you will not be seen or burdened with this using BioRankings Analytics software. So I want to start off by speaking about data sparseness in metabolomics, or missing data. Now, what we have here on the screen is a generic workflow for a researcher that he has decided to do some combination filtration and integration methods in order to overcome the missing data problem, right? And this is one of the ways, one of the many ways. I'm not saying that there's anything wrong with it, but this is one of the many ways. And I can imagine a researcher sitting in front of a computer screen, picking what MZ slices to, and this is, decision that really leads to downstream effects in your workflow. Getting to the point that it makes reproducibility difficult, because if you keep in mind that this might be the workflow that researcher A takes to get to his result, then researcher B might decide that this combination of MZs might not be the adding additional MZs. And you will see through our software, there's no deciding of combination filtration integration. It's all in the method that we have decided to apply. Alignment is a big issue in metabolomics in general, not just untargeted metabolomics. Now, this paper does a very fine job of showing this illustration of what some features might look like when your data is not aligned, right? And they provide their solution to it and what it should look like when it's aligned. This getting back to the point that, we are all providing research and tools and development to for us to use, but they're not taking into account the entire workflow, and these tools are being built independent. I'm sorry. And it goes back down to, it's researcher driven, and it's based on the method that the researcher uses and the parameter that the researcher uses to make the decisions in this particular case of alignment. The application that we have chosen to use for our analysis in our software does this automatically already. This paper did a very fine job of pointing out corrections that your experiment might need. You might have problems with batch effects, misalignments, or some type of instrument drift. Now, these are generic representations of what the signal might look like, but they did a very fine job of illustrating that. I say that to point out that we have built this into our software. You will actually be seeing a batch effect example later on in the presentation, where we have automated and simplified how to correct for the batch effect when analyzing your untargeted metabolomic experiments. So keep in mind those workflows that I showed at the beginning of the presentation. They were very hands-on, they were very detailed. You did huge amounts of work. You've gone through your workflow, you've aligned data, you've interpolated, extrapolated, whatever you might have needed to do in order to get those features at a point where you actually can do some comparative analysis or some kind of statistics. And now what I have on my right-hand side on this diagram is some generic box plots. It's an illustration to represent some features that have been chosen after a researcher has gone through his workflow for two different groups that he's interested in comparing. Now, the problem with this is that when you've already gone through that workflow, made decisions that has affected your downstream analysis, now you have this final set of features that you're willing to calculate p-values on. Well, now you're in a space typically known as hypothesis, as the results are known, harking, because you're really going to be testing what you want to test. You selected features you want to test. And just by looking at these box plots, you can tell which ones you can expect a significant difference on. With that being said, BioRankings software will test everything that you upload to it. It will find a feature in the entire data set, and it will test everything that it finds in the entire data set. So a little bit about our method and our application and our solution. We're using what's called functional data analysis, FDA. FDA is a method that was built to analyze data that comes in forms of functions or graphs. And that's what we're doing here. We're viewing LC-MS untargeted metabolomic data in forms of functions. Now, what you have to your right-hand side is a 3D representation of the FAHKO experiment. You have the 12 mice, 2 groups, 1 wild type, 1 knockout. Of course, you can't tell the difference between the 2 groups here, but this is to illustrate the fact that we can, after conversion, we can see this data as functional format and avoid those pain points that we highlighted at the beginning of the talk. By us applying functional data analysis, we are no longer having to deal with the workload of interpolation, extrapolation, or combining MZs, as you saw in the previous workflow examples. So you might be familiar, might be more used to seeing something like this in your outputs, as opposed to the previous slides. Now, what we have done is just take that data and looked at a specific region, 3271 to 3272. And after conversion to functional format, we can clearly see our 2 different groups. Our knockout mice are represented in the black curves, our wild type mice are represented in the red curves. And pretty easily we can, see 2 different features at 2 different RTs there. So with that being said, our application of functional data analysis onto untargeted metabolomic experiments opens the door for functional linear modeling, which then allows us to do multivariate statistics, allows us to leverage the information from your experimental design and do those kinds of analyses that take into account the batch effect, do analyses that take into account repeated measures or any that you can really think of. And you'll be seeing examples for the batch effect in the later on videos. Our software does currently offer repeated measures analysis, and we'll have additional videos outside of this conference available for those later on. So when can you use BioRankings Analytics software? It's really after you have collected your data, put it through the LC-MS machine, and you have one of the current file types that we allow in the software. You upload your files, and on average, we're seeing people get results back within 2 hours. So we are really proud of what we have built, just to highlight again some of the key things that we have done, and you will see later on in the videos. Eliminate those pre-processing steps. You will no longer have to sit in front of a computer screen. In our experience, we were hearing some research, we're sitting in front of computer screens, slicing and dicing MZs for days to get to that final step in the workflow to do comparative analysis. And we make reproducibility possible. As you saw previously, I gave you a scenario where you can have researcher A, researcher B, researcher C take different approaches, and it makes reproducibility impossible. With that being said, what I'm going to be showing you today in the demonstration videos, we're going to be showing you how to register. It's a pretty simple process. We're going to be showing you how to upload some untargeted metabolomic data. We're going to be showing you how to analyze that data in a group comparison analysis in an ANOVA approach. We will also have that COVID example that takes into batch effect. Depending on where we are on time, it looks like we might be good on time. We will be getting to a multi-omic analysis where we're using classification regression tree application to analyze microbiome and cytokine data. So with that being said, I'm going to jump in straight into the first video. Let me know if you guys cannot hear the audio? I'll adjust
Hello
it.
and welcome to today's video.
We can hear
Today
you.
I will be showing you how
Thank you.
to register for BioRankings Analytics software. I begin by showing you our home page. Now for you to get here, just go to your browser and type in analytics.biorankings.com. Brings you to our home page. From here you have access to all of our software. Just go to the bottom of the page. You can find links to our social media and our privacy policy and terms and conditions. Scroll up to the top of the page because today we want to sign up. So let's click sign up for free. Let's bring up our sign up for free page. We have some information to fill in. Let's type in my name. My organization is BioRankings. My email address for today, I would say temp4@biorankings.com. I will select the password. Confirm my password. And click I'm not a robot. Get verified. Final step, click sign up. Now you get the login page. So let's verify that we successfully created an account. And now we have successfully created an account and logged in, and we now have access to all of the software on BioRankings Analytics platform. That concludes today's video. Thank you very much.
So as you guys saw, it's pretty simple, and you'll see this approach being taken as you see the later on videos. We're taking an approach that we're trying to simplify things for the researcher, not only from an analytical perspective, but from the point you get to our website. So what do you get for signing up for BioRankings Analytics software? Well, you get access to all of our untargeted metabolomic data analysis tools. You can analyze microbiome data. As you probably saw, is currently in the beta stage. And as I highlighted in the first slide, this is all work based on collaborating and talking with researchers like you. So the more we collaborate, the more we talk with people, the more we can add to this platform and the more you have access to. So you will get access to those additional features as we continue to build it out. But you get access to the BioRankings team as well. So that's really with any questions you might have, running analysis, interpreting results, managing your data, really taking any feedback, any questions you guys might have. We are here to build a platform really that helps you guys get to your results faster. So you've signed up, you probably want to run your analysis now, but first you need to upload some data. So what you need to do first is upload the data. You will need to go to our homepage, go to the upload data page that will look something like this, fill out the information. But to really get the whole concept, we put together a video for that, and I will start that now.
Hello and welcome to today's video. Today I will be showing you how to upload your untargeted metabolomic data to analyze it through BioRankings Analytics Software as a Service website. I start off by showing you two windows. On my left hand side there is a folder with a spreadsheet and some MZML files. I will give further detail about those files in a minute. To my right hand side, I have the BioRankings Analytics Software as a Service homepage. Keep in mind, I'm already logged into my account. But for you to get here, all you have to do is go to your browser, type in analytics.biorankings.com, and this will bring up our homepage. So to begin, we want to find the metabolomics beta drop-down menu at the top of the page. We'll click that, click on your data. Now you can see that my profile currently hosts two data sets. However, we want to upload a new one. Let's find the upload new data blue button at the top of the page. Let's click on that. This brings up our upload data page. At the top of the page, we have some information. that tells you what file type our software as a service currently allows. We currently allow .cdf, .mz XML, .mzml, and .mzdata.xml. However, if you have a different file type, please do not hesitate to contact us. We can analyze your data outside of this tool. The next bit of information are some sample data sets that we offer to users in case they want to try out the software but do not have their own data. If we scroll down, the first piece of information required is information of our metadata file. So we begin by naming it, and the next step is to browse and find that metadata file. Now, as you can see, my file is named covars.csv. That's why I named it metadata file underscore covars, but it can be named anything. To briefly explain what's in your metadata file, for this particular example, I have two columns. My first column is named file name. My second column is named treatment. I have a total of 16 samples. And these 16 samples could have received 1 or 3 treatments. Please note that this file name in the spreadsheet matches up exactly to what's in my folder. So let's close that out. We will not save. The next step in our browser is to upload our MZML files. We will click Browse, go to the folder that holds our data, select our files we want to upload. I have a total of 16 files. You can see that this box gets filled with the 16 files I have selected to upload. Just click the blue button. It says Upload Files. If successful, you will get a green box here that says Success and the total number of files you have uploaded. The last step is optional. However, we do recommend it if you plan on running several analyses. So this is for our demo dataset. The last step is to click this button that says Submit a Data Upload. And that brings you to our Start Analysis page. But let's verify that our data has been uploaded to our profile. So let's go back to the top of the page, Metabolomics Beta drop-down menu. Click that. Click on Your Data. Brings up the Your Data page. You can see that the first entry now is the data set we have just uploaded, demo dataset. The timestamp. If we click on Manage Metadata, this gives us details about our data set, our attached metadata file, and from here you have the ability to attach additional metadata files in case you have additional variables you want to analyze. Let's close that out. This icon under File List lists out the files you have uploaded to your data set. This icon under the Share column allows you to share your data with collaborators. All you have to do is input their email address. The only requirement is that that collaborator must have a profile on the BioRankings Analytics platform. Let's close that out. The last piece of information is this delete icon. You have the ability to delete a data set if you wish. Just mark this yes, delete this data, and click the red button. However, I do not want to delete this data. We will close this out because we will be using this demo data set in the next video for analysis. Thank you, and I will see you in the next video.
So as you guys saw, we try to make it pretty straightforward. However, if you have any questions, do not hesitate to contact me after the conference, and we'll get you squared away. Really want to touch on the data you just saw being uploaded. because that's the data we're going to be seeing in the next video being analyzed. We had 16 samples. As you saw, they could have received treatment ATP, TGF-beta, and the remaining were control samples. We're interested in knowing if there are any metabolite difference between these groups, and that's the analysis you're going to see being run. I want to highlight that this data was sourced from the metabolomics workbench, and it has been modified for this demonstration. If you want full access to it, you can get it through that reference. With that being said, we're going to see how to analyze this data.
Hello and welcome to today's video. Today I will be showing you how to analyze the data we previously uploaded in our data upload video. I begin by sharing two windows. On my right hand side is the BioRankings Analytics homepage. I'm already logged in. However, for you to get here, just go to your browser and type in analytics.biorankings.com. To the left, I have the data we previously uploaded. As a reminder, let's look at our metadata. We had 16 samples. Those 16 samples either received treatment ATP or TGF-beta, and the remaining samples were control samples. Today, we will be running a group comparison analysis. We are interested in seeing if there's any metabolite differences between these groups. Let's close up our file. We will not save. Let's go back to the BioRankings Analytics homepage and find the drop-down menu for metabolomics beta. Click on Start Analysis. This brings up the Start Analysis page. At the top of the page, you will find some information of what you can expect to receive after running your analysis. You can expect to receive a spreadsheet that will contain every feature identified in your experiment. Through our user interface, you can expect to see a peak plot for every feature identified in your experiment. If we scroll down on the page, we currently offer three types of analyses: a group comparison, what we will be running today, a group comparison with adjustment for a batch effect, and a repeated measures. Please look at our additional videos for those types of analyses. So let's select group comparison. The next step is to select our data source. We want to select demo_dataset. The next step is to select our metadata file. Now I only have one metadata file attached to my dataset, so it's pre-filled for me. The next step is to select our metadata columns. Just click on that. We currently allow users to have up to 3 columns in your metadata file. Today I only have treatment. I will select that, click Save Columns, and you will get this prompt. You selected treatment for your analysis. Let's scroll down on the page. The next step is to name our analysis. The next step is to set a minimum intensity. This value must be between 1,000 and 1 million. I will choose 6,000. Now what this means is that my output will not have any features that had an intensity less than 6,000. The next step is to set a minimum feature width. This must be between 2 to 30 seconds. I will select 6 seconds. Now what this means is that my output will not contain any features that were not at least 6 seconds long. The next step is optional for a targeted comparison analysis. If you have a list of targeted metabolites you wish to compare to BioRankings findings, just provide us with a spreadsheet that has at least 2 columns. One column that identifies MZ and one column that identifies RT, and we will complete that comparison for you and provide you the results. We don't have that today, so let's click on Start Analysis. After some dataset checks, you will get this message if successful: data successfully sent. You will receive an email indicating that we have received your data and is currently being analyzed. Once your analysis is complete, you will receive an additional email notifying you that you can now review your results. And that concludes today's video. Thank you very much.
So as I mentioned earlier, guys, that's what we're trying to key here, really the simplicity of how to run your analysis. So what have we done? We have run the analysis for all the data, so that's running in the background. What that's going to do is identify every feature in that data set you have given to us, then do that test that we mentioned earlier. We're interested in see if there are any group differences in those 16 samples that uh, we saw in the metadata file. And what you did not see is handle in the background through the software all of those pain points that we previously highlighted in the presentation. No more slicing and dicing, no more alignment on your end. It's all handled by our application of functional data analysis. So in the next video, what you guys are probably interested in is the interpretation of the results.
Hello and welcome to today's video. Today I will be showing you how to access and interpret the results from our previous data analysis run video. To begin, I am sharing two windows. On my right hand side is the BioRankings Analytics homepage. I'm already logged in. However, for you to get here, just go to your browser and type in analytics.biorankings.com. To the left of that, I have the email that I received when my analysis was complete. At the top of the email, you will find the title of the analysis. Below that, you will have a link to access your results in the interactive space. Towards the bottom of the email, you have a link to download a PDF that will contain the top 100 features identified in your analysis. To the right of that is a spreadsheet that will contain every feature identified in your analysis along with any statistics. Let's go back to our BioRankings Analytics homepage because we can also access our results through here. To do that, we need to go to the top of the page, find Metabolomics Beta drop-down menu, then click on your results. Now you can see our analysis is at the top of the page. Let's click on View Results. Let's maximize this window. And this brings you to additional information about this analysis. Who ran it, when it was run, additional run information, the data that was used. You have another link to load the interactive space. I have already loaded this for us. We will look at that shortly. Below that, you have a receive via email link in case you need to receive your results through email again. This direct download button, if we click it, you get access to those same files. However, you have two additional files you can access. The first one being this whole peak data.csv. This file will contain the data for every sample, for every feature identified in your analysis. This is useful for any post-hoc analysis you might need to do. The last one is a targeted comparison CSV file that will be available if you ran that targeted comparison analysis. Let's look at some results. We start off by showing you the peak table on the top left corner of the page. In the middle, you have your peak plot. To the right of that, you have your annotation table. At the bottom left corner, you have a box plot representation of the feature. To the right of that, you have a spectrum plot for the feature selected. Now let's walk through an example or two. I have highlighted the first row. We are focused on peak number 31 that was found at MZ307 with an apex at 676.5. This feature was found to be statistically different with a p-value of less than 0.001 and an effect size of 90.21. An effect size tells us the magnitude of the difference between groups. Let's go to our peak plot. Our control samples are represented in the red color. The shading around the bold curve is the 95% confidence intervals. Now our two treatment groups, TGF-beta and ATP, are represented by the green and blue curves respectively. We can clearly see that our control samples have higher intensities than our two treatment groups. This particular example did not have any HMDB matches when comparing it to that database. If we scroll down on the page, the box plot representation of our peak. To the right of the page, you will have peak number 31 highlighted in yellow, and you will see additional bars on the spectrum plot if they had roughly the same RT. And in this case, we did not. Below that, you will have additional statistics for that peak that is highlighted or any peaks that had roughly the same RT. Again, we did not have any in this case. Let's look at a different one. Let's look at peak number 3. And let's look at some different settings. Let's say I'm interested in seeing the adjusted p-value in the post-hoc test results. Now, you have the ability to do so through the settings menu. I'm not interested in intensity match, so we'll remove that. Unchecking it will remove that column from your peak table. This menu towards the bottom of display options will change the visuals for our peak plot and our box plots. We'll close this out. And now we can see that peak number 3 found at MZ117 with an apex of 187.5 was again statistically different even after adjustment. And our post-hoc test is finding that our control samples are different from our 2 treatment groups. And we clearly see that our red curve is higher in intensity than our 2 treatment groups. This particular feature did have 6 candidate metabolites. If you click on the first one, it will take you to the web page for that particular metabolite. Let's go back to our user interface. We scroll down again. The box plot for our feature. A spectrum plot, again, did not find any other features that have roughly the same RT. And that will conclude today's video. Thank you very much.
So those videos have been kind of short, guys, due to knowing the time constraints of the conference. But if you see anything in there that you were interested in that I did not touch on, feel free to contact me after the conference. So what have we done and what are the advantages to testing out BioRankings Analytics software? Where automatic identification of every feature in your data set that you give to us based on MZ and RT. We test everything. We find candidate metabolites comparing it to HMDB by default. Currently in our beta stage, we have worked with some clients where they were interested in other databases. So if you are interested in finding candidate metabolites in a different database, we have the ability to do so, but we will have to do that off the beta version, and we can just do that on a one-on-one call. It's pretty straightforward for us to do that. With that being said, again, our leverage of functional data analysis leads us to do things like batch correction. And that's the COVID study that I mentioned earlier that we worked with Dr. Gary Patty at Washington University School of St. Louis with. So we're going to be seeing that analysis next.
Hello, and welcome to today's video. Today, I will be showing you how BioRankings Analytics software can automatically adjust for the batch effect in your untargeted metabolomic data. To begin, I share with you 2 windows. On my right hand side is the BioRankings Analytics homepage. I'm already logged in. However, for you to get here, just go to your browser and type in analytics.biorankings.com. On my left hand side is the metadata file that I have already uploaded to my profile and have analyzed and will be showing you results for shortly. I would like to point out that we work with Washington University School of Medicine in St. Louis, the Gary Patty Lab, to analyze this data, and these results are currently in print and will be published soon. So, in this experiment, we had 324 samples that were tested for COVID. Again, each row represents one of the 324 samples in this metadata file. Column C represents our group column. These 324 samples were given a label of either severe COVID, mild COVID, or they tested negative for COVID. We also found out that these samples were collected in different batches. So we have added column B to our metadata file named batch. And this batch column identifies when samples were collected. So let me explain briefly a couple of samples. We can see that sample in the second row was collected in batch 1 and it had a severe case of COVID. Sample on row 7 was collected in batch 2 and had a severe case of COVID. Sample in row 20 was collected in batch 1, had a severe case of COVID. And by adding the one column that contains the batch information, BioRankings Analytics software will automatically adjust for this effect. So let me briefly show you how you would go about analyzing your data that needs a batch effect adjustment. So let's go to the drop-down menu, go to Start Analysis. Again, I've already run this analysis for us, so I will not be running this. But the difference now is that you will select Group Comparison with Batch Adjustment. And now BioRankings Analytics software will analyze your data, adjusting for the batch effect. As I mentioned, I've already run this analysis. Here's my result page. I've already loaded our interactive space to review some results. Let's look at some. I currently have highlighted the peak identified at MC 246.07. It was found to be statistically different with a p-value of less than 0.001, and that's actually the adjusted p-value. Our post hoc test shows that the severe samples are different from the negative samples, and our severe samples are different from our mild samples. Let's look to the right hand side of this, our peak plot. Our severe cases of COVID are represented by the green curve. The shaded part represents the 95% confidence intervals for each one of the groups. Our mild cases of COVID are represented in the blue curve, and our negative cases of COVID are represented by the red curve. But we clearly see that our severe cases of COVID have higher intensities than both our negative and mild cases of COVID. This particular feature did not have any candidate metabolite matches when comparing it to HMDB, so we have a blank table. Bottom left corner, we have a box plot representation of our feature. To the right of that, we have our spectrum plot. Again, we have our feature highlighted in yellow that we have currently selected, and next to it we show that there are two other features that have roughly the same RT. Scroll up, look at some different features in this COVID study. Let's look at the first one. Our severe cases in this particular feature are in green. Our mild cases are in blue, and our negative cases are in red. We found after adjustment the p-value was less than 0.001, and our post hoc test found that severe samples were different from both our negative and mild samples, but also our mild samples were different from our negative samples. And in this particular feature, we do have some candidate metabolites we can look at. Again, if you click on the link, that will take you to the HMDB website for that particular metabolite. Let's go back to our results. So with that, one additional column in your metadata file You can now automatically adjust for the batch effect when using BioRankings Analytics software. Thank you very much, and that concludes today's video.
So what did we just do, guys? We automatically adjusted for the batch effect that has several different ways of getting done. But through BioRankings Analytics software, it takes one column in your metadata file. Just tell us what batch your sample was collected in and let the software do the work. So to really emphasize what our software is built behind is built behind using the experimental design information from your data and really the statistical modeling approach that we're taking, the functional data analysis approach. So what else can bioranking software do? Well, I had left it open for questions, but I still have a lot of time left, looks like it. So I'm going to jump into the multi-omic analysis application CART that we have available on our beta software at the moment. As I mentioned, this will be using microbiome and cytokine data. It's a supervised clustering of your microbiome experiment. So with that being said, we put together a small video for that, and let's look at it.
Hello and welcome to today's video. Today I will be showing you some results for a CART analysis that I have run through BioRankings Analytics software. As you can see, I'm already at the home page logged in. However, for you to get here, just go to your browser and type in analytics.biorankings.com. After logging in, it will bring you to our home page. As I mentioned, I've already run our CART analysis, but you might be asking, what is CART? Well, CART is short for Classification and Regression Tree Analysis. It's an exploratory analysis tool that can be used to discover any microbiome groups that might be associated with your metadata. So let's say, for example, you've collected microbiome data in your experiment, but you've also collected other information, for example, clinical information, demographic information, or even other omic information. This tool can help discover any microbiome groups that might be associated with those variables. It's a supervised clustering of the microbiome analysis. So, before I show you any results, let me show you how to run a CART analysis. You want to go to the top of the page, find Microbiome Beta drop-down menu. Under CART, find Start Analysis. This will bring you to the CART Start Analysis page. At the top of the page, you will find information about what you can expect to find in your results when running CART. Beneath that, you will find some of the requirements needed to run CART. Below that are some sample data sets that can be used to run CART in case you don't have your own. This is the data that I will be presenting results for today. Beneath that, you will find a link to the paper that has published this data and can provide further detail about the method being applied in our software. As I mentioned, I've already run the analysis and I've loaded our interactive space. So let's look at some results. When running CART through our software, you can expect to see something like this for your results. I want to start today by looking at the metadata file tab. My analysis today had 128 samples. Each row in this table represents a different sample. Each column in this table represents a different variable that was used in my analysis. In my case, each one of these columns is a different cytokine. Again, I am interested in finding out if any of these variables are associated with any microbiome groups in my experiment. So let's go back to tab number 1, and these will be the visuals we provide for the results in your analysis. On the left hand side is a tree plot. On the right hand side is a bar chart plot. As I mentioned, this is an exploratory tool, so you have the ability to use this slider to visualize your results at different levels. You can see that the tree is being pruned as I move the slider. So today, let's look at a small tree where we can easily interpret these results. Starting on the left-hand side with our tree plot, we had 128 samples. Our analysis found that the cytokine leptin with a threshold of 1937 was the most important when discovering microbiome groups. So all of my samples with a leptin level less than 1937 went to terminal node number 2, and that was a total of 68 samples. All of my samples that had a leptin level higher than 1937 went into node number 19. My analysis then found that the cytokine IFNG with the threshold of 66 was also important in finding microbiome groups. So my samples that had high leptin but low IFNG went into terminal node number 20, 41 samples. Terminal node number 27 had my remaining samples. These samples were made up of high leptin and high IFNG, a total of 19 samples. To my right hand side, each one of these bars represents one of my terminal nodes. Each one of these colors represents a different taxa that make up the microbiome composition for these groups. So, if we look at the first bar for terminal node number 2, we can see that these were for the samples that had low leptin, and we can see that the microbiome composition for this group had high Bacteroides, about 42%, compared to our samples in node number 27 that were high in leptin and high in IFNG had very low Bacteroides, about 16%. And with that, you are now able to discover any microbiome groups that might be associated with your metadata. That concludes today's video. Thank you very much. Hello.
So as I mentioned, guys, we kept these videos short. There was a lot of information I was not able to within that and that particular now more results in to provide researchers with as much of the information and results that we can, because we know like to post hoc analysis and do additional to it. So if there's something in particular that you were interested in, just don't hesitate to me. This is contact me or anybody at BioRankings. And I would like to open it up for questions if there are any.
Thank you, Carlos. Lots of informative videos. We learned a lot of cool tools for people to try. I think there's a bunch of questions I'm going to email you, but a couple of questions we can take. One is from our Thusi. She's asking, Carlos, when you are correcting the batch effect, you don't use any PBQCs or QCs in different batches?
No, we are only using the information from the statistical model. So our approach is to identify the variance introduced from the batch effect. If you can tell me what batch effect your sample came from, I can tell you how much variance was attributed to that effect. Same concept when it comes to collecting data from multiple subjects in the same experiment. If you tell me you have repeated, if you collected multiple measurements from Carlos, I want to remove that variance from the result. I don't want to introduce that variance into the result and possibly introduce false positives. So we can identify that variance, remove it, and provide you with that final end result, which is what you're seeing in the user interface.
Thanks,
Yeah.
Thank you.
Yeah. One more interesting question from Golf is how BioRankings tools compare to XCMS or MetaboAnalyst for handling the data?
Well, I can't speak to those tools. You know, we saw them and we know that we had researchers, as I mentioned, that had concerns, that had gaps in their workflows, and we got funded huge amounts of money to develop the software, right? So we know that those tools are great for certain things. For example, targeted analysis for people who are computationally inclined, who can sit there and write a script and do, do everything that our software is doing and write code to get it done. But we also know that there was a real discrepancy when it came to getting work done and time to result. And that's what we brought to the table, really, the automation piece and really leveraging our software engineer power to really get these analysis done quick.
Got it. Thank you, Carlos. I think there is a couple of questions from Maz and Georges. So I'm going to email those to you, and I've already
Awesome.
emailed. So yeah, so that can help you. But with the best interest of time, we are way over time for the entire event. So I would like to thank you very much, Carlos, for your time and staying up this late. Your
Thank
next
you
day
for
has
having me. This was a great opportunity.